All right, so with all of the specimens, what they need to be looking at is the sensory organs. All right, so we have the eyes, we have the nares, which are on the back. The nares is a little bit more complicated than we were originally probably looking at. We have both an excurrent and an incurrent hole. They need to make sure that they understand which one's the excurrent, which one's the incurrent. The other big sensory organ that they're going to have to be looking at, especially for the cat, is the ears. With the cat, the ears have the pinnae. They're also going to have vibrissae, and we're going to talk a little bit more about the ear structures in just a little bit. So they've already looked at the lateral line system running down the entire length of the shark. This lateral line system, if we were to look a little bit more in depth, there's actually a canal that's running down on the inside of the skin. That canal is filled with fluid from the outside environment, and it's connected to the outside environment via pores. Anytime there's a pressure change within the outside environment, maybe from you kicking as you're swimming away from jaws, it's picking up on those pressure changes by water coming in and out of those pores and into that lateral line canal. So they need to understand how the lateral line canal works. All right. This lateral line canal will run all the way up and will go up along the head as well. So when we first skinned the shark, there's going to be canals running in a whole bunch of different directions up here. That lateral line system becomes more complex. That lateral line system also runs on the ventral side of the rostrum. All of those black dots that are along the rostral portion or the ventral portion of the rostrum are connecting to that lateral line system. These are called the ampullae of Lorenzini. They're found on both the ventral side, and they would also be found on the dorsal side. These are once again picking up on pressure changes, but they're also picking up on electrical stimuli within the environment. So as you're kicking your feet through the water, you're also creating an electrical discharge. Same with a fish that might be trying to swim away from the shark. And the shark is using this as more of a sensory organ as to where the prey is, or where maybe a predator is, or something else is within the environment. All right. All of those are going to be connected to the facial nerve, which we're going to get to in just a little bit. So the facial nerve is what's picking up on all of that sensory information. All right. If we were to look at the two spiracles on both sides, directly in between the spiracles on the dorsal side of the shark, so the actual skin of the shark, you would see two other pores that are much bigger than these ampullae of Lorenzini. Plus, the ampullae of Lorenzini are down towards the rostrum. These two are endolymphatic pores. Right? Endolymphatic pores have endolymphatic canals connected to them, and that's going to be running deep. Okay. Michelle, can you get the cast of the concha cranium over there? So take a step back from the shark real quick. If we were to look at the cat ear, or any mammal ear, so we'll pretend it's the cat ear. The cat ear then has a hole that then goes down into the ear canal, and our ears would work the same way. At the base here, you would have the eardrum. You need to know it as the tympanic membrane. Just a thin membrane. And then this is going to be connected via three structures. You can remember it as MIS, M-I-S. You have the malleus, the incus, and the stapes. And these are going to be connected to a bigger structure that's more of a cavity. That's called the vestibule. On the far end of the vestibule, you have a snail-like shell structure, and this is the conchae. My spelling might be a little off. When you pick up on sound vibrations at the tympanic membrane, those are transferred over to the vestibule. Those are then picked up by the conchae, and this is where sensory neurons will transmit those to a bigger nerve. Okay? That is called the cochlea nerve off of the cochlea, the conchae. Okay? Over here, you have canals that run off. And those canals run along different planes. You might have another one running in another weird convoluted plane. But ultimately, all of these canals, these three canals, are running at different planes. These are called the semicircular canals. Okay? If we look at our shark cast, these red structures are located within the otic capsule. Those are the semicircular canals of the shark. Just like the shark has them, so does the mammal have them. Where we have a vestibule on the mammal, you have a sacculus on the shark. This is the empty cavity where those semicircular canals are emptying into. All right? All of these semicircular canals are doing the same thing. They're maintaining equilibrium. Within these semicircular canals, this was one of the semicircular canals, you have cilia. And attached to the cilia are neurons that then come together to form a big nerve. Okay? Within the semicircular canal is fluid, and as that fluid moves, it moves the cilia. So if you hold your head straight, and then you move your head to the side, you move the fluid within the semicircular canal, which moves the cilia, and then that gives your brain and your body a sense of where you are in space, hence equilibrium. Okay. This on the mammal brain is picked up by nerves, and that's the vestibular nerve. So that when these two nerves come together and form one larger nerve, that's the vestibular cochlear nerve, which is one of the cranial nerves. Fun fact of the day, which I tell everybody, so we'll throw it into the video too. But there's a lot of blood capillaries that are surrounding this. That's why on the shark cast it's red, because there's a bunch of blood capillaries sitting there. If someone were to drink too much alcohol, the alcohol will diffuse into the semicircular canal, and because of the viscosity difference, like oil and salad dressing, it'll swirl and it will move the cilia, and this is why the room spins if somebody were to drink too much. It's not because the room is actually spinning, it's because you're moving the cilia within your semicircular canal. Interesting. Okay. So that's how semicircular canals work, and it's going to be the same for both the cat, the mammals, and the sharks. Everything's the same. Okay. Just because we're not going to get to it later, within your handouts, you need to know a few more terms for the cat. You need to know that this is the pinnae, that's the outer ear structure. This right here is the external auditory meatus. This is all within the otic capsule, these structures. Going from the otic capsule to the inside of the skull, you have another canal, and that would be the internal auditory meatus. And that's how that nerve is going to reach the brain. Okay? So you need to be able to follow that pathway of those nerves. All right. So that covers, one last thing. The endolymphatic pores that we just talked about. Those canals are going to come down and they're going to connect with the sacculus, which was where the semicircular canal is also connected with. This is providing more information as to where the shark is in space.